Are you in the cell of an organism that is currently undergoing growth, development, repair, or produces offspring from one parent, better known as asexual reproduction? If so, are you a chromosome that is sick of staying in the same place as your sister chromatids? If these qualities apply to you, try the cell cycle, the new way to escape being in the same cell as your sister chromatids and start your own cell. Here is Kenny, who has tried out the cell cycle and loved the results, and Dr. Marley, who is an expert in cell division.
I'm so glad that I tried out cell cycle and recommend it to all sister chromatids out there who felt just like me. With this program, you can go through interphase, mitosis, cytokinesis, and voila, you have a whole new cell to start by yourself.
The cell cycle lasts as long as the cell is living because it's the repeating set of events in the life of a cell. Dr. Marley, could you tell us more about the first step of mitosis?
A lot of people mistake interphase to be the first step in mitosis, but in reality, interphase is just the time in between cell divisions that is broken up into three parts: G1 phase, the S phase, and G2. G1 is simply the phase where the cell will grow to be full size because the offspring cells are half the size of the original cell. G1 actually stands for the amount of time after cell division and before DNA replication.
I remember from my experiences that during the S phase, the cell's DNA is synthesized, or copied. And then during the G2 phase, the cell I was in had prepared for cell division.
If you decide to give cell cycle a try, then you would go through interphase first, then enter mitosis. The first step in the 4 steps of mitosis is prophase. Prophase starts with the shortening and tight winding up of DNA into rod-shaped chromosomes. Because during the S phase you were copied, you are now connected to a sister chromatid by the centromere.
During prophase, the walls around me, which were the nuclear membranes, broke down and disappeared. Then 2 pair of dark spots appeared next to the nucleus.
2 types of spindle fibers are called connected core and polar fibers. The connected core fiber is attached to a dish-shaped protein called a connected core that is found in the centromere region of each chromosome. And these fibers extend from the connected core of each chromatid to one of the centrosomes.
So it's the polar fibers were the ones that extended across the cell from centrosome to centrosome without attaching to the chromosomes.
After prophase is metaphase. Metaphase is the step where you'll be most clear when going through mitosis, so people will be taking pictures of you in this stage to make karyotypes. Also in this stage, the kinetochore fibers will move you to the center of the cell and hold you in place.
After metaphase came, anaphase and I finally separated from my sister chromatids. We separated at the centromere and slowly moved. Centromere first, towards opposite poles. I was finally an individual chromosome. After anaphase came, telophase came. After I reached to the opposite pole of my sister chromatid, the spindle fibers disassembled and we returned to a chromatin state where we were tightly coiled. A nuclear envelope formed around each set of the chromosomes and a nucleolus formed in each of the cells.
Right now, for an extra $200, we include cytokinesis, which is the cytoplasm dividing during telophase. In animal cells, cytokinesis starts with a pinching inward of the cell membrane midway between the 2 poles. In plant cells, the vesicles from the Golgi apparatus join at the midline of the dividing cell to form a cell plate, from which a cell wall forms, dividing the cell into 2 new ones.
The area of the cell membrane that pinches in and eventually separates the dividing cell into 2 cells is called the cleavage furrow. The cleavage furrow pinches the cell into 2 cells through the action of microfilaments. Offspring cells are almost equal in size and each gets an identical copy of the original cell's chromosomes and about half of the cell's cytoplasm and organelles.
I was unsure if I wanted to go through the cell cycle because I didn't know if it was safe or controlled. But soon I learned that there are proteins which regulate the progress of the cell division at certain checkpoints. Certain feedback signals from the cell can trigger the proteins to start the next phase of the cell cycle. The 3 main checkpoints are the cell growth checkpoint, the DNA synthesis checkpoint, and the mitosis checkpoint.
Mitosis phase of the cell cycle is absolutely safe to use because the cell growth checkpoint doesn't let unhealthy cells divide and only lets safe cells move on to the next phase, S, where DNA is copied. Also, if you're in a cell that cannot divide or else it won't do its job, mitosis makes sure that those cells pass on to the G0 phase and never divide again.
DNA synthesis is a checkpoint when the DNA repair enzymes check the result of DNA replication. If the checkpoint is passed, the proteins will signal the cell to begin the processes that allow the cell to divide methodically.
The last checkpoint in the cell I was in went through the mitosis checkpoint. When the cells passed, protein signaled the cell to exit mitosis and I entered the G1 phase again.
The prokaryotic cells go through binary fission, which is a division of prokaryotic cell into two offspring cells. The DNA is copied, which results in two of the same chromosomes attached inside the inner cell membrane. Then a new membrane begins to form between the two DNA copies. The cell grows until it reaches about twice the cell's original size, and as a new material is added, the growing cell membrane pushes in and the cell is constricted in the center. A new cell wall forms around two membranes and split into two independent cells.
For a prokaryotic cell, each contains one of identical chromosomes that result from the copying of the original cell's chromosome.
Try the cell cycle. It's a fun, easy way to start your own cell.